In this video, we discuss a review of the evidence for pain mitigation in piglets undergoing routine procedures such as castration, tail docking and teeth clipping. This project was funded by the National Pork Board and has two parts. The aim of the first part was to summarise what evidence is available for the effect of pain mitigation interventions, and the second part describes the process for developing recommendations based on that evidence. One of the areas to focus on for this review was the outcomes identified that could be used to assess pain mitigation. The review panel decided that those outcomes measured within 24 hours, shown here, were those that are most likely to reflect the effect of pain mitigation strategies. This list includes behavioural and non-behavioural outcomes. One of the important aspects was that this list was created prior to the conduct of the review. So the experts were asked to identify what outcomes were important before knowing what outcomes were measured. This is important because often studies are done using outcomes that are perhaps easy to obtain but not necessarily highly important. And so identifying these outcomes prior to the conduct of the review is important. On Table 3, you see we have data about the studies that were included in the review, and most of them relate to castration. There are 42 and 10 related to tail docking. Other characteristics of the studies are also included here. And you'll see we were fortunate enough to be able to include papers that were not written in English. One of the things that we used frequently in this paper was forest plots. Here we have Figure 2, which is a forest plot, and there's an enormous amount of information you can obtain from a forest plot. First of all, starting on the left-hand side, what we have here is the interventions that were used for the animals. You'll see that we have isoflurane and combinations of carbon dioxide with oxygen. So there are several interventions. Next across we have the data on the mean of cortisol, which is the outcome that was measured. So each line of data represents one study. After presenting the mean for the intervention group, we have the standard deviation. If this information is not presented by the author, as in the first case here, then it is not included. The next column is the total, which represents the number of animals that were included in that arm of the study. Moving from left to right, we then have the mean, the standard deviation, and the total for the control. The weight here refers to how much weight is given to the summary, to that evidence in the summary effect measure, which we'll discuss later. Each study is summarized by an effect size. Cortisol is a continuous outcome, and so the way we measure the effect size is subtracting the mean of the intervention from the mean of the control. That gives us a mean difference. If the mean difference is 0, then that indicates that the mean is the same for both groups, which would suggest that the intervention had no effect on the outcome cortisol. So the mean difference is called the effect size, and we also calculate the 95% confidence interval for each mean difference. Finally, moving across to the actual plot, we now have a plot of the mean difference, whereas previously we had just presented the data. The vertical line here presents the line at which the mean difference is 0. You can see that for studies where we were able to obtain means and standard deviations in the number of animals, we're able to calculate a summary effect, which is represented by the green box, and a 95% confidence interval for that effect measure. And how precisely we know that effect measure is indicated by the width of the 95% confidence interval. Notice here that we have 3 studies for CO2O2. If we combine data from those, we get the summary effect, which is indicated by the black diamond. Each study contributes information to the calculation of that black diamond, and that's the amount of information it contributes is given by the weight, and that's a function usually of the sample size. Some studies don't have a summary estimate because the data were reported in a way that we couldn't extract information from them. When reading the summary effect, the vertical axis of the black diamond indicates the point estimate of the summary effect, and the points of the horizontal axis of the black diamond represent the edges of the 95% confidence intervals. On the forest plot, effect sizes, which are green dots or green boxes, that are on the left-hand side indicate a favourable outcome for the intervention. That is, cortisol was less in the intervention group than the control group, which would suggest that it is favourable. You can see for the CO2O2, the effect is very variable. In one study, it favoured the intervention. For the others, it favoured the control, and another estimate is very close to 0. So when we calculate those together, the estimates all combine to suggest that there is really little evidence that there was a large difference or an effect of the intervention CO2O2 on cortisol levels. When you look across all of the protocols, you'll see that some are favourable, some are not favourable, and some are very close to 0. This is referred to as heterogeneity. This heterogeneity prevents us from combining data across the protocols because there's evidence that the protocols have different effects. This can be contrasted by looking at another table, another forest plot, which is Figure 5. Figure 5 presents the information with cortisol again as the outcome. However, this time the intervention is non-steroidal anti-inflammatories. What you can see is that most of the effect sizes are on the favourable side, and there is more data for each of the interventions. So in this example, we have less heterogeneity. And this decrease in heterogeneity played an important role in Part 2 when we were developing recommendations for the use of intervention therapies. Notice again here that some studies didn't report completely, and so it wasn't able to calculate a summary effect size. But there were several studies for meloxicam and several studies for ketoprofen, and it was possible to calculate a summary effect size for those interventions. Both these papers are available on open access, and I encourage you to look at the entire paper to understand the scope of the work and how it should be interpreted.